In lab mice, 18 months marks the biological threshold of late middle age. At this stage, the standard physical markers are easy to see: a hunched posture, thinning, unkempt fur, and a noticeable lack of movement. But here, the contrast is stark. This second mouse is the exact same age, yet it remains active, well-groomed, and maintains a straight, healthy posture. This difference was produced by a new combinatorial drug called SRN-901. Researchers used a clinical frailty index to track this physical decline. While the control group in black saw their health markers deteriorate, the mice given SRN-901 saw that progression slow by 70% over 128 days. This chart shows the incidence of age-related tumors in both groups. The rate dropped from 19% in untreated mice to 13.2% in the SRN-901 group, a relative reduction of nearly 30%. These results suggest that targeting aging pathways directly can preserve the structural health of the body, even as the organism grows older. This Kaplan-Meier chart tracks survival. The red line representing SRN-901 shows a 33% increase in median remaining lifespan compared to the control group. Statistically, this represents a 46% reduction in the overall hazard of death. To put that in perspective, look at the other lines. These represent highly publicized single-molecule treatments like NMN, NR, and Rapamycin. While some showed benefits, none achieved the survival increase seen in the SRN-901 group. This leaves us with a significant question. Why did this multi-compound approach succeed, where decades of research into single-molecule drugs have hit a ceiling? Biological aging functions as a cascade failure. When one system slips, the added stress damages the next, leading to a system-wide collapse. Treating this by targeting a single gear rarely stops the overall breakdown. SRN-901 is designed as a combinatorial therapy. It merges several discrete compounds, including NAD+ precursors and autophagy enhancers, into a single intervention. The goal is to engage multiple interconnected aging pathways at the same time. By hitting several targets at once, the drug attempts to neutralize cellular threats across different biological fronts. Because aging is a system-wide failure, researchers are finding that it requires a systems-level medical response. To identify exactly which biological processes were being affected, researchers used RNA sequencing to map the cellular changes triggered by the drug. This chart maps the genetic pathways coordinated by the drug. Notice the downregulated NF-kappa B pathway, suppressing chronic inflammation. Similarly, reduced mTOR signaling slows cellular overgrowth. Conversely, yellow bars indicate accelerated protective mechanisms like DNA repair and the p53 tumor suppression pathway. The drug also boosted mitophagy, a critical internal routine where cells identify, engulf, and dissolve their own damaged structures before they cause further dysfunction. This coordinated effort, suppressing inflammation while accelerating repair and waste removal, creates a cellular environment that is much more resilient to decay. These genetic changes translate directly into the physical chemistry of the blood. By sequencing the metabolome, researchers could see how the drug prevents metabolic drift as the mice age. This chart shows a heavy upregulation of glutathione metabolism. Glutathione is a master antioxidant, and boosting its production builds a biochemical defense against oxidative stress. This recalibration shifted the internal chemistry of the older mice. Their blood metabolite profiles were adjusted to more closely resemble those of younger animals. This internal reset appears to be the mechanism behind the youthful appearance, reduced frailty, and increased survival observed in the study. The data from this study highlights the potential limitations of the single-molecule approach that has dominated longevity science for years. The survival gap between SRN-901 and single-target drugs suggests that treating aging as an interconnected web is more effective. This study provides evidence that a combinatorial approach is a viable path for significant extension. This multi-pathway strategy also showed secondary benefits. The data revealed a reduction in biological markers associated with neurodegeneration and systemic decline. Mitigating age-related decline is becoming an engineering challenge. The evidence suggests the most effective solutions will address the entire biological machine at once. Focusing on healthspan means prioritizing independence and physical activity in our later years. While human clinical trials are the necessary next step to confirm these findings in people, researchers now have a clear blueprint for targeting the underlying mechanisms of aging. We are seeing that extending life and physical resilience is a solvable problem, provided we use a systems-level approach to the biology of aging.